It's come to my attention that the previous review article that I published on YouTube, which was intended to help people understand the potential mechanisms by which ivermectin works, it was designed to elucidate those mechanisms so that the average person could understand it. Apparently, I didn't do a very good job. People complained. They said that it was a little bit too complex for the average person. So what I'm going to do today is to try to make that easier to understand. Upon review of the information, it has come to my attention that it was a little bit complex and that there's a certain amount of information that it assumed that the viewer knew that, and it just went with it. And if you don't have that pre-existing information, then it makes it harder to understand. So what I'm going to do today is I'm going to try to summarize what that video had in it, but give you more of the background information so that you understand it better. So after having seen today's presentation, hopefully you'll have a better idea as to how we think ivermectin works to fight cancer. Okay? So I'm going to start like this. Ivermectin is a really cool drug, in part because it's very cheap right now, and in part because of the low amount of side effects. That's probably the reason it won the Nobel Prize, was because it's an anti-helminth medication, an anti-parasite medication with low side effects. Let's say you find out that you have a roundworm swimming around in your gut, and you don't like it. Let's call him George. George is bothering you. You're losing weight that you weren't trying to lose because this little sucker's in there eating your calories. Some people might like that, but there are a lot of people that just don't like the idea of a worm crawling around inside them. So you want to kill that sucker. Well, poison works really well when it comes to killing things. So of course you could drink a bottle of cyanide and that would do the trick. Unfortunately, there would be collateral damage. You don't really want to deal with the side effects of a bottle of cyanide. So you want to find a poison that's going to kill that worm, but not you. This is where, the old medicines they lacked, they were less than desirable. You take a drug like albendazole. It worked. It does help with helminth infections or worm infections, but there were some significant side effects. Caused a little GI bleeding, which resulted in black tarry stools, bleeding gums, blood in the urine or stools, chest pain, painful or difficult urination, painful pinpoint red spots on the skin, sore throat, sores, ulcers, and white spots on the lips. So, yeah, quite a number of side effects. And that's because the worm physiology and your human physiology have a lot in common. So it was difficult to direct a poison that would just kill the worm and not get you. Now, this is where ivermectin shines. Ivermectin specifically targeted a pathway that isn't used very much, if at all, by other animals other than the parasites. One particular pathway, one particular channel that the parasites have that people and our farm animals don't have is something called the glutamate chloride channel. All right? And in response to glutamate, a chloride channel opens up, and when chloride has a negative charge, when it rushes into the cell in high amounts, it causes something called hyperpolarization. In other words, for motor neurons, it will paralyze that motor neuron. So if you're using your muscles for things like breathing, you stop, okay? And that results in death for those parasites. So for organisms that infect you that have these glutamate chloride channels, you add ivermectin, and ivermectin binds to that site on the channel. It opens that channel up, chloride rushes into the cell, and it's neurotoxic and therefore death. And it doesn't take a very high dose for ivermectin to work as an anti-parasite drug. 3 to 6 milligrams for just a few days, and boom, you're good. This was revolutionary for people in the United States that had farm animals. Now, first, it was for people in countries where you had a high incidence of worm infections. This was great because finally we have a cheap drug with low side effects that is really good at kicking those worms in the head. So, great drug, very wonderful. And if the story had ended there, ivermectin would still be a great drug. But it turns out this particular drug has many other properties. And some of these properties make it helpful for not just treating helminth infections, it's also useful for treating viral infections and even cancer. Now, I'm not going to get into the antiviral stuff because that's a whole nother chapter. A lot of people knew about that and they're familiar with ivermectin because of its efficacy with regard to things like HIV and COVID. I'm not here to talk about that. I'm here to talk about the use of ivermectin for the treatment of a variety of cancers. So right now it's not approved. It is not FDA approved for the treatment of cancer. However, we have a number of studies, preclinical animal models and few clinical trials, which show that it does appear to have anti-cancer effects. And I'm going to talk about that right now. Ivermectin has been studied for its potential anti-cancer effects primarily through inhibiting specific signaling pathways that are often dysregulated in cancer cells, but less critical in healthy cells. And so that's what we're taking advantage of. What are the differences between a cancer cell and a healthy normal cell? Now, as you may know, the more aggressive a cancer is, the more different the cancer is from your normal human cells, the easier it is to kill. You know, if you have a cell that's growing very, very rapidly, it's outrageously doing mitosis, then you can target your therapy to zap those cells that are doing ultra-high mitotic rates. So that's a difference, and that's what you want to do. You want to find an anti-cancer therapy that is specifically going to hit your cancer cell without whacking your normal cells. So that's what we want to take advantage of. The first signaling pathway, there are 4 signaling pathways that I want to discuss. So ivermectin inhibits these pathways which are present in the cancer cell, but not so much in normal cells. And for this reason, we can use ivermectin in physiologic doses without a bunch of side effects that can be aggressive with the cancer cell. Now, I will say this. The dose, here's where we're at right now. Not a lot of research has been done to the point where we can identify what dosages are necessary for these specific cancers. We are so far behind here. I mean, where we should be is we should have already had clinical trials looking at each cancer type and saying, okay, what is the best dose to use? What doses are toxic? What doses are too little? It would be great to have that information. We don't have it. Well, why not? Why hasn't the money been spent for these trials? We know that it works, you know, it works in the lab, you know, it works on animals. Why don't we have this human trial, this human data? It costs a lot of money. It costs millions of dollars. Nobody wants to spend that money for a drug that is cheap. Nobody wants to spend that kind of money for research on a drug unless there is a multi-billion-dollar payoff. That's the problem. So that's where we're at. I wish I had better news, but unfortunately, keep that in mind. Now, we do know that the doses used to fight parasites are itty-bitty and very low compared to doses necessary for killing of cancer cells. Okay? So, I mean, keep that in mind. So let's talk about those pathways. The first one, the WNT beta-catenin pathway. WNT signaling has emerged as a fundamental growth control pathway. It's important in embryonic development, cell cycle regulation, inflammation, and cancer. Now, this pathway this signaling pathway is especially overactive in cancer stem cells. So, by blocking this pathway, you are targeting cancer stem cells. What are cancer stem cells? All right, stem cells are the cells that when they make babies, they give rise to the actual cancer cells. They're the mother of the cancer cell population. And so they stay in the deep tissues, all right? And they do nothing but make babies. And when they make babies, they're producing baby cancer cells, right? Now, when you use chemotherapy or radiation, it's not that difficult to whack the grown-up daughter cells, the cancer that you observe, but what you're not getting often are the cancer stem cells, which are deep in the tissue. Okay? Now, this particular pathway, the WNT catenin pathway, this is active in those stem cells. So by whacking this particular pathway, by inhibiting this pathway, we are making the stem cells sick, promoting their death. Okay? Now, when you disrupt the WNT signaling, what you're doing is promoting the degradation of beta-catenin. And when you disrupt that, this beta-catenin is a protein that drives the cancer to proliferate. And what does cancer cell proliferation means? It refers to mitosis, the ability to divide and make brand new cancer cells. And we don't want cancer that is actively dividing. Cancer cell proliferation is bad. That means our cancer is getting worse. It's growing and growing. And so by disrupting the WNT signal and degrading the beta-catenin, you are stopping that driving force. So you're slowing down the cancer cell's ability to divide and make new cancer cells. Okay? We are also reducing the cancer's ability to survive, because it needs that pathway for survival. Now, why doesn't it damage us? Because healthy cells do not rely on the WNT signaling pathways. Very, very, very little. Our healthy cells are healthy, and so this is a pathway that unhealthy cells have to rely on. Most cancer cells are metabolically sick. Their mitochondria are not doing very well. They are not able to do oxidative phosphorylation very efficiently, and they are reliant on these alternative pathways because normal pathways are closed to them. Okay, so we're taking advantage of that. We're saying, okay, you're using this cheater pathway over here, this WNT signaling pathway, which we don't need. Now let's whack that so you're taking their crutches away. All right, that's what you want to do. There's another pathway, the AKT-mTOR pathway. Ivermectin suppresses the AKT-mTOR signaling cascade, which regulates cell growth and metabolism. Cancer cells often depend on this pathway for rapid proliferation, kind of like the previous pathway. So they need it to rapidly do mitosis and make new cells, while normal cells, once again, are less sensitive at therapeutic concentrations. In stem cells, this is another one of those pathways that is overly active. Now, when ATK-mTOR pathway is active, what it does ultimately is it stops the cell from appropriately committing suicide. Okay, when you refer to the process of apoptosis, think of the word pop, like as in explode and die. Apoptosis refers to cell death. When cells become sick, when they're not able to make energy and breathe and do living stuff, when they're sick, what's supposed to happen is you flip a switch and it tells the cell, I'm supposed to die because I need to die. I need the natural killer cells to come here, and I will die by suicide, and then the natural killer cells will take me out and they'll eat me, and my nutrients will be available for the healthy cells. That's what's supposed to happen. So apoptosis is the process of cell death, programmed cell death. When cells are sick, they're supposed to die. And then you end up with autophagy. Autophagy is when those healthy cells are able to eat the sick cells so that the nutrients don't go to waste. Okay? That's what's supposed to happen. In cancer cells, unfortunately, when the ATK-mTOR pathway is active, it allows the cell, even though it's sick, to avoid flipping the suicide switch. It was supposed to commit suicide, and it didn't. It's still alive. It's still making babies, even though it's sick. We don't want it to make babies. You're making sick babies. We don't want those sick babies. No, we want healthy. We want you to die, and we want your nutrients to be given to the healthy cells. What are you doing here? We don't want your cancer in the body here, right? And so when ivermectin inhibits this AKT-mTOR pathway, you allow the cell to realize it's too sick. It's supposed to commit suicide. It's supposed to do apoptosis. And so when you inhibit that pathway and the cell realizes, I'm supposed to do apoptosis, that's what happened. And you increase apoptosis of cancer cells. So what you're doing is you are pushing the cancer cell, because it's like barely hanging on anyway because it's sick, but it's not dying because it's healthy enough to hang on because of these pathways, because AKT-mTOR, it allows it to go on. By blocking that pathway, you push it over the edge and you drive it to suicide and autophagy, which is what you want. So that's what you want. You want to see those cancer cells start to shrink and die, and that happens through apoptosis. So ivermectin inhibits the AKT-mTOR pathway. Another signaling pathway is the purogenic pathway. Now this is one of the more complicated ones, and I'm going to try to explain this very simply, because as you see, a lot of words here and a lot of acronyms and everything, so I don't want to get into that too much. The thing you need to remember about the purogenic signals, specifically take a look at this diagram here. Look at this top of your screen, where you have a couple of channels. You have the P2K7 channel, the P2X7, P2X4. All right, those are ion channels, and when ivermectin is doing its job, basically cancer cells have an excess of these P2X7 receptors. And when you hit those receptors, when you modulate those receptors which are involved in purogenic signaling, it promotes cancer cell survival. When you hit those, when you inhibit those receptors, you end up with an alteration in ion flux. And so what ends up happening is your cell swells and swells and swells, which identifies it as a sick cell, and you promote cell death. And so this is something that induces apoptosis. So specifically, by inhibiting the P2X7 receptors, you promote apoptosis through a different pathway than the purogenic signaling. Than the AKT-mTOR pathway. Many cancer cells have excess P2X7 receptors, and ivermectin makes those cells swell up and die. All right, and it spares healthy cells with lower receptor levels. Now, it's not that healthy cells don't have any P2X7, but far, far less, and so they are much less affected by ivermectin. All right, and now number 4, the YAP1-TAZ pathway. So the YAP1-TAZ pathway, this is a pathway that is especially expressed in things like liver tumors. Okay, there are other tumors, but predominantly things like liver tumors. And when this pathway is active, it promotes the migration and infiltration of the cancer cell. All right, it impairs contact inhibition. What is contact inhibition? With healthy cells, as they're growing in your petri dish and a cancer cell meets the side of the petri dish, or it meets another cell, it will stop replicating. It'll turn off mitosis. Hey, stop having babies. We've reached somebody else. I'm touching my neighbors, and I want to make sure there's enough food for everybody in the room here. So if I keep having babies, somebody's going to starve. So I'm going to stop reproducing once I hit a wall or I hit another member of my species. So if I bump into somebody else, or I bump into a wall, I bump into another cell type, it's time to stop growing. And when the YAP1-TAZ pathway is going boogity, 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 as it does in cancer cells, you blow by those restraints. You look at that red light and you say, screw that, I'm going on anyway. And so you keep dividing, you keep differentiating, you keep proliferating. And so ivermectin impairs that pathway to stop that aggressive movement. When you take here you see an example of some cells on a petri dish there, and you notice all the green cells, those are tumor cells, and they're very high in the YAP-TAZ expression. And so ivermectin reduces the activity of YAP-TAZ as part of the Hippo signaling pathways, and therefore you are dropping tumor growth, you are preventing tumor metastases as well. So you're stuck, because when a little bit of the tumor breaks off and goes to another part of your body, it can't metastasize there if density dependent or contact inhibition is still working, because if it's bumping up to a different cell type, it'll say, hey, there's not room for me to grow. I've got neighbors here. I need to be nice to my neighbors. I don't want to just move into this neighborhood and start having babies if there's not enough room. And when YAP-TAZ is overactive, it doesn't care. It just keeps having a bunch of babies, right? So ivermectin blocks that. So it helps to prevent metastases and excess proliferation. Normal cells, they're not regulated. They don't have a problem with excess YAP-TAZ. So it's not going to affect your normal cells nearly to that same degree. Okay? So that gives you an idea as to those are 4 pathways that are inhibited by ivermectin that are not expressed to large degrees in normal cells. Okay? Now, talking about differences between normal cells and cancers, we really have to discuss one of the things that it's not really a new finding. We've known this for a long time, but only recently have we started to really wake up and pay attention to this truth. And that is that most cancers, the overwhelming majority of cancers, suffer from mitochondrial dysfunction. In fact, we believe, many of us believe, that the primary damage, the primary problem with cancer cells is not the mutation, but rather mitochondrial dysfunction. Mitochondrial dysfunction leads to an increase in the production of reactive oxygen species, also called free radicals, and those free radicals are like radiation, and those damage your DNA, and those DNA damaged areas result in mutation and can activate oncogenes and give rise to various tumor types. But it started with the mitochondrial damage. See, I did my PhD is in the field of cancer genetics. I studied lymphoid neoplasia, looking at cytogenetics and molecular biology of leukemia, lymphoma, and so forth. That was my PhD area. And the premise of my doctorate was incorrect because it was based on the assumption that the primary insult in cancer is the mutation. And it turns out that is probably not true, that the primary insult was the loss of intact mitochondria, the loss of effective oxidative phosphorylation. It's not that cancer cells can't do any oxidative phosphorylation, just that it is severely limited. And cancers have to find alternative means of generating energy other than using the electron transport chain in oxidative phosphorylation. Here in this particular slide at the bottom, look at part B down there, and you'll see you notice on the left-hand side, it shows glucose going to pyruvate, and then that pyruvate is entered into the tricyclic, the TCA cycle. And ultimately, you see the production of ATP and NAD and NADH, and that's called the respiratory chain, and that's how normal cells get their energy. You take glucose, goes to pyruvate, that pyruvate gets dumped into the mitochondrial respiratory tree, and through a variety of enzymes in the mitochondria, you make ATP, adenosine triphosphate, and that's the source of electricity for all the cells in your body, including all your healthy, I should say, that gives you energy for all the cells in your body. Now, what happens in cancer cell is that oxidative phosphorylation breaks down. There is a problem with the electron transport chain, so that we are making less ATP from the mitochondria. All right? And our energy then, instead of going through the tricyclic pathway, we end up going through glycolysis. We go through fermentation. And so that's what you see here, that glucose, you're getting pyruvate, but you're also getting lactate, and you're just going boogity, boogity, boogity through anabolic pathways. And so it's a less efficient way of getting energy. But that's what you see happening with these cancer cells. Now, because of that, because you're not getting energy the normal way, that puts you at a disadvantage, and so you have to use these alternative pathways in order to stay alive and to function. So you can still operate, but you're using a much less efficient means of generating energy. Okay? So that's important to understand. We've known this for a long time. There's a Dr. Warburg that I think it's like from the 1920s. Yeah, and it's called the Warburg effect. And he discovered this way back then, that cancer cells are more likely than not to generate energy through glycolysis rather than oxidative phosphorylation through the mitochondria. Okay? One of the things that metabolic therapies take advantage of is the fact that cancer cells do glycolysis rather than oxidative phosphorylation. They can only burn 2 fuels, glutamate and glucose. That's it. And so by going on a glucose-restricted diet and taking certain glutamine blocking drugs. There is right now very good evidence that that is an incredibly effective way of starving the cancer early on. Once you start using chemotherapy, you flood the area with glutamine and glucose, and it makes it harder for metabolic therapies to be effective. Okay, but that's the basis. It takes advantage of the fact that cancer cells, they don't do oxidative phosphorylation. Your normal cells do. Your normal cells don't need any glucose. They don't. They can make glucose from protein. They can make glucose from fat. They don't need glucose. Whereas the cancer cells absolutely need glucose, and if they can't get glucose, they can use glutamine. And so that's what metabolic therapies are based on. Okay. Now, ivermectin is particularly toxic to cells with mitochondrial dysfunction. All right. For 4 reasons, maybe 5 reasons. 1, mitochondrial membrane potential disruption. Ivermectin can affect mitochondrial membrane potential by inhibiting complexes in the electron transport chain or modulating ion channels. That's something that we talked about a minute ago. Cells with pre-existing mitochondrial dysfunction, often characterized by impaired membrane potential or reduced APT production, are less able to compensate for this additional stress, leading to heightened toxicity. So what does that mean? Well, these cells are not doing well. They are impaired. They are not able to do oxidative phosphorylation. They're hanging on by the skin of their teeth, doing glycolysis, and they're like teetering. And so when you disrupt the membrane potential, because one of the things that generates that takes a lot of energy is maintaining all of these pumps. You got your sodium pump, you got your calcium pump, you got potassium pumps, and it takes ATP, it takes energy to keep those pumps going. Those pumps break down, you die. Okay? So when you use ivermectin and you disrupt those pumps, then you throw that balance off and it makes it easier to die. And so that's one of the reasons why ivermectin is toxic to cancer cells, but not so much to healthy cells. Okay. Oxidative stress amplification. Mitochondria are key regulators of reactive oxygen species. Ivermectin may increase reactive oxygen species production by disrupting mitochondrial respiration in cells with dysfunctional mitochondria, which already struggle to manage oxidative stress. This can exacerbate damage, triggering apoptosis or necrosis. I mentioned before, cancer cells, they can do a little oxidative phosphorylation, not very much. Even the little bit they do can help. Now, what happens in these sick cells, in a healthy cell, when you make ATP, 95% to 99.5% of the time, you're mostly making just ATP, but you're always going to get some waste product. And in healthy cells, from 5% to 0.5% of the time, those waste products are going to be free radicals, reactive oxygen species, so superoxide radical, hydroxyl free radical. Those are like radiation, and they cause damage, okay? And ivermectin is now it is somewhat damage to mitochondrial function, but if you have healthy mitochondria, it's like they can just blow it off. It's like, that's nothing. We can handle that because we're making a million ATP, and you cost us a couple hundred. We can handle that. However, in cells that are already sick, in cells with already damaged mitochondria, you just jack that damage all the way up. And so now you are making even more reactive oxygen species. And so you're flooding the area of the cancer with radiation. Okay? That's what these reactive oxygen species, those free radicals, are like radiation. And the area of the cancer is now even sicker. You've amplified oxidative stress using ivermectin. Okay, but normal cells, you don't have to worry about that so much because they can handle this. All right, dependence on mitochondrial pathways. Cancer cells or other cells with mitochondrial dysfunction due to mutations or metabolic stress often rely on altered mitochondrial pathways for survival, such as upregulated glycolysis or specific signaling cascades, such as the AKT-mTOR. Ivermectin's inhibition of these pathways hits these cells harder as they lack the metabolic flexibility of healthy cells. And I pretty much discussed that already, so it's just kind of stating what I already said in a different way. Okay? Apoptosis sensitivity. Dysfunctional mitochondria are often primed for apoptosis due to altered Bax BCL2 ratios or cytochrome C leakage. Ivermectin's ability to induce mitochondrial-mediated apoptosis, such as via the P2X7 receptor modulation or reactive oxygen species, disproportionately affects these cells, as healthy cells with intact mitochondria can better resist such triggers. So again, stating it a little differently than I said earlier, but we talked about the apoptosis sensitivity. And again, this is something that is specifically going to be a problem with your cancer cell, not so much with your normal cells, because the cancer cell already has these dysfunctional mitochondria and they're primed for apoptosis already. I mean, as I said, they're right on the edge of suicide and you just push them over. Why is ivermectin more toxic to cells with mitochondrial dysfunction? It attacks genes that are more expressed in cancer stem cells. Okay, there are a number of genes that are, I mentioned this earlier, but there are a number of genes that are specifically highly expressed in cancer stem cells. Those stem cells are the most important cells in the cancer. When you use chemotherapy or radiation and you kill the bulk of a tumor, that's great, but if the stem cells are still alive, those are the cells that are making most of the babies. That's where the cancer came from. And if the cancer stem cell is still alive and ticking, it was going to make the cancer all over again. And so you're going to be going back and forth, back and forth. Oh, I'm getting better. Oh, I got the cancer again. Oh, I'm getting better. Oh, the cancer's back. Okay, stem cells. So several genes, as I mentioned earlier, are highly expressed in cancer stem cells, and ivermectin attacks those. We talked about those earlier. So that's cool. So we already know that ivermectin attacks pathways that are present in stem cells. We didn't talk about some of the other, they call stemness genes that are specifically associated with cancer stem cells. And they describe NAN, N-A-N-O-G, SOX2, and OCT4. They maintain cancer stem cell pluripotency and self-renewal. And so, in other words, they make sure, like within a particular cell population, you have multiple cell types, and they all come from those stem cells. And the ability of the stem cells to renew themselves is present there. And so these particular genes are involved in the stem cell's ability to survive and replicate. And when, especially in breast cancer cells, it's been shown that ivermectin significantly downregulated these genes in cancer stem cell enriched populations, even at concentrations that were relatively low, 4 micromoles. That's a pretty low concentration, with minimal effects on normal cells. And healthy stem cells rely on different regulatory networks for their function, so they're not as sensitive to ivermectin. Okay? So those, hopefully, I know it's a little bit complex, but I hope I've given enough background information so that you have a better understanding as to why ivermectin may be effective for many cancers without causing significant side effects to the host normal cells. Okay? So hopefully that makes sense. Right now it's not FDA approved for cancer, but if you're a clinician out there and you want to do some clinical trials, or if you're in a pretty bad way and you want to talk to your doctor about, giving it an off-label try, hopefully you have some information that you can educate yourself on and maybe educate others. Okay? Thank you for watching.